Good morning, students. Today we will discuss seed germination. All the seed contain an embryo and have their own food supply. The embryo consists of a plumule, epicotyle, cotyledon, hypocotyle, and radicle. Plumule. The plumule includes the young primordial leaves and growing point of the stem. Next is epicotyle. The epicotyle is the portion of the stem above the cotyledon. Next is cotyledon. The cotyledon are the seed leaves used for food storage. And next is hypocotyle. The hypocotyle is the portion of the stem below the cotyledon. Next is radicle. The radicle is the young embryonic root and root tip. Next is germination process. So what is germination? Seed germination is the process of reactivation of metabolic activity of the embryo, resulting in the emergence of radicle, that is root, and plumule, that is shoot, thus leading to the production of seedling or young plants. Germination stages, as shown in the figure. The process of seed germination involves several consecutive and overlapping events like absorption of water. Water absorption is also called as hydration phase, in which the water is absorbed by the process of imbibition and osmosis by the dry seeds, which soften the seed coat and other covering, causing hydration of the protoplasm. After imbibition of water, the seed swells and seed covering rupture, which help the protoplasm in resuming metabolic activity with the activation of enzyme. During this phase, the seed coat acts as limiting factor and its rupture increase water uptake. Water enter the seed through micropore and helium. Next is synthesis and activation of enzyme. After hydration, enzyme activity begins very quickly. Activation of enzyme is partially from the reactivation of stored enzyme and partly from synthesized enzyme during germination initiation process. The hydrolytic enzymes convert complex food material into simpler form, which can be readily translated and absorbed by the embryo. The oxidative enzymes are involved in respiration and releasing energy from cell division and growth. Next is cell elongation. Hydration, synthesis, and activation of enzyme help in elongation of cell, which result in the emergence of radicle. Emergence of radicle is the first visible symptom of germination, which result from the elongation of cell rather than from the cell division. We can say that Under the favorable conditions, emergence of radicle may take place within few hours, as in non-dormant seeds, or few days after seed sowing. The emergence of radicle is considered as the end of first stage, that is activation stage. Next is translocation stage. Food material, that is fat, carbohydrates, protein, are stored in endosperm or in cotyledons. These compounds are converted into simpler form and are translocated to growing point of the embryo. The process of conversion of different type of food material which reserve in the seed, for example, fat and oil, they are converted enzymatically to fatty acid, then sugar, protein to amino acid, and nitrogen starch to simpler sugar. All these conversions are regulated by metabolic activity of specific enzyme in a proper sequence. Next is seedling growth stage. In this stage, we can say that the development of seedling take place from cell division in different growing point of the embryo, which followed by the expansion of seedling structure. The cell division in the growing point and subsequent cell elongation are two independent processes taking place in seedling. As the germination proceeds, the structure of seedling soon become evident. So with this, we have completed stages of seed germination. Next is types of germination. First one is epigynous, as shown in the figure. The hypocotyle elongate and raises the cotyledon above the ground surface. It is known as apical germination, as shown in the figure. This is very common in beans, gourds, castor, tamarind, onion, etc. As shown in the figure, the other type, that is hypogyle, in which the epicotyle elongate and hypocotyle do not raise the cotyledon above the ground. It is mostly found in gram, pea, custard apple, lotus, maize, etc. Next is factors affecting seed germination. Many factors affect seed germinations. First one is water. Water is the most important factor which affects the seed germination. Seed become active only after the absorption of water. The germination percentage or the rate usually depend upon the water absorbed by the seed. In general, we can say that seed germination do not occur at moisture below 40 to 60%. But in certain plants, germination can be occur in the low moisture conditions. The rate of water uptake is influenced by the many factors like nature of seed, prevailing temperature, availability of moisture content, etc. It is also known fact that impermeable seed coat more often act as barrier for water absorption, and similarly water absorption is higher at higher temperature up to a certain limit. But it is all time essential that optimum moisture should be available in adequate amount to initiate the process of germination. The excess moisture may also be harmful to the seedling. Next is temperature. Temperature play important role in initiation and regulation of seed germination. It affects germination percentage and germination rate of the seeds. Every plant species have certain range of temperature below or above which germination process is stopped. We can say that the optimum temperature is the one at which the largest number of seeds germinate at the highest rate. The temperature requirement of seed varies from species to species. For example, seeds of cool season vegetables like cabbage, broccoli, celery may germinate at or below 4 degrees centigrade, whereas the seed of warm season vegetable, tomato, cucumber, pumpkin, they germinate freely above 10 to 15 degree centigrade. So the optimum temperature range for the seeds of most of the food plant is 25 to 30 degree Celsius. Next is aeration. Germination. Germinating seeds respire at very high rate. Thus, adequate supply and exchange of gases below germination medium and the embryo is necessary for rapid and uniform germination. Subsequently, the seed growth. The oxygen requirement of different seeds vary widely, but some seeds germinate freely at the oxygen tension below, that is the atmospheric concentration. The supply of oxygen to the germinating seed can be limited either by seed coat restrictions or excessive water supply to the growing media. Similarly, hard soil crust, which is formed by excessive irrigations or heavy rainfall, also inhibit the seed germination processes. Next is light. Seeds of certain plants have specific requirement for light and it will not germinate in the absence of light. Light intensity have relatively less effect on seed germination, but photo period and wavelength have more effect on the seed germination. In general, the seed germination in blueberry, strawberry, raspberry, etc. is influenced by the light. In contrast, seed germination in phlox, pansy is retarded by the light. The seed in which germination is promoted by light are called positively protoblastic, and in which the germination is inhibited by light are called negatively photoblastic seed. Next is temperature and light interaction. Both temperature and light combination have great influence on seed germination. It is particularly true with the flowering annuals, which have been classified into 9 different groups on the basis of temperature and light requirement. Next is salinity. Salt present in the soil may also prevent or retard germination of some cases. Salt become more problematic if the growing media is light and irrigated with poor quality of water. Frequent light irrigation results in salt deposition on the upper surface of the soil. The problem of soil is acute in area having high salt in water, and this problem can be avoided by using the soils and water with low concentration of salt. Next is pathogens. Pathogens also affect the seed germination, like damping of seedling is the most important example of in which the pathogen completely destroy the young seedlings. So proper care must be taken to avoid pathogen organism in the growing media. Next is tool injury. It is likely that germinating seed or seedlings are injured by some tool during intercultural operations, which may also result the poor germination of the seeds. Next is seed coat barriers. Seed coat of some species is impermeable to water due to the presence of some microsclerate cells which inhibit the water. As a result, non-dormant embryo is unable to develop and emergence due to moisture stress. Next is rest period. Post-extraction rest period is sometimes responsible for the poor germination. Example is fig. Next is treatment to facilitate germination. Number one is by soaking the seed in water. It is common practice to soak seed in the water before sowing, which promote germination by softening the hard seed coat, activate the enzyme and diluting the effect of imbibition. Next is stratification. Stratification, also called as moistening, which is practice of exposing the seed to low temperature for considering period after ripen the embryo, which is helpful in the germination of seed. Next is scarification. It is the process of engineering, breaking, softening the seed covering to make them permeable to water and improve the gas exchange. Next is hormonal treatment. We can use cytokinin GA3 to break seed dormancy. Next is chemical treatment. We can use thiourea and other chemical to break the seed dormancy. Next is dry storage. Freshly harvested seeds of some species fail to germinate until or unless stored for some period. To facilitate germination in seed of such species, the seed should be dried at 40 degrees for 3 days, then stored for some time before sowing. Last is light exposure. Light is necessary for germination to take place in positively photoblastic seeds. With this, we have completed Seed germination.